The overall goal of this methodology is to describe how a transcription factor of interest regulates the microRNA transcriptome.
This method can help answer key questions on whether transcription factors that modulate transcription of protein coding genes are also involved in transcription of microRNAs. The main advantage of this methodology is that it integrates data mining, laboratory data, and bioinformatic analysis to provide a comprehensive description of microRNA genes transcriptions that is dependent on the activity of transcription factor of interest.
Though this method can provide insight into the interaction of microRNA gene transcription and activity of the transcription factor studies, it is important to remember that this is a descriptive method and confirmation of such an association should be done using additional assays.
To begin, open a genome browser, such as the University of California Santa Cruz Genome Browser, in order to extract chromatin immunoprecipitation sequencing data. Once on the site, go to the top menu, select Tools, and open the table browser. Enter the specifications, as shown here, into their respective boxes, and then select Get Output to extract the table. Save the output as a text file, then import the file into a spreadsheet. Once in the spreadsheet program, sort the name column and filter the results for a transcription factor of interest, such as STAT2. Then, copy the list of microRNA promoters.